Okay, wonderful. Welcome, everyone. Thanks so much. If you can turn on your video, it would be greatly appreciated. It's always great to see faces. If not, of course, we understand. Today we have a great roundtable, and Whitney will introduce Dr. Stefan Pinter from UConn. But before we do that, we thought it would be great for many of you. I definitely enjoy meeting AP heroes and just getting to hear from parents about what their experience has been like. So we're hoping for those that are not families affected on this call to also just get to enjoy for a few minutes and meet some of our kiddos as we do these monthly. So Talena has been amazing, her and her husband, Megan. We were able to meet in Colorado last summer, and we wanted her to introduce her gorgeous AP hero and tell us a little bit about herself, her family, and her AP hero.
Hello, my name's Talena. This is Delilah. She is 26 months old. We are from Portland, Oregon. And Delilah was officially diagnosed when she was 11 months old. She has AP inversion duplication deletion, and then she also has a uniparental disomy of her chromosome 16, and she has Duane syndrome as well. And those are all her official diagnoses, and also a bunch of vision stuff that I won't bore you with rattling off, but she just mastered crawling right around her second birthday. She does OT twice a week, PT and speech every other week, and feeding therapy once a month. And and then of course I do all of those things all day long during play and eating and bath time. Well, you know how it is. I would say that she's just always progressing. She's always getting stronger. She's always learning new skills. I don't think she's ever regressed. She's plateaued definitely lots of times, but she's great. Anybody have any questions? Sorry, I don't know what else to say.
Yeah,
that was great. Thank you for sharing.
Feel free to ask questions, as some of them, some of the parents are on these calls. So it's really just to the researchers and the scientists, clinicians, to, if you had a wonder, if you had a thought, this is why we're doing this with families, so that we can be able to expand the curious minds here. I know that next time we'll have Silje, who's in transit right now over in Europe, and Yanni, who's been a wonderful AP mom as well. They're just going to wave and say hi to all of us, but we'll wave to you, Silje. I see the smile. That's awesome. And they've tried a lot of really amazing practical therapeutic approaches of really addressing the groundwork that we all need in terms of our development. So they've been a great resource to families, and I'm sure to many of us that are just parents in the field. So it's amazing all the things that we can learn about neurodevelopment, just hands-on being a parent. Thank you so much, Talena. It's so good to see Delilah. And Whitney, go ahead without further ado.
Thanks. It's really nice to meet you guys. Our presentation today is by Stefan Pinter, Dr. Stefan Pinter. He's an assistant professor at the University of Connecticut. His lab in the past and currently has worked on a number of chromosomal syndromes. So he's worked on trisomy 21, Turner syndrome, and he's also been developing a number of tools to probe these different syndromes in different ways. So working on tools like EXIST, And today he's going to tell us about some CRISPR tools that he is developing to study AP, as well as tell us about some of the other work in his lab. So I'll let him take it from here.
Whitney, thank you very much for the great introduction. And thank you for this opportunity to share with you our work and the types of things that we're thinking about. We tried the share screen right before we got on, but now I'm not seeing my PowerPoint. Let me see whether I can just.
I'm not staying here. I have the
Share my desktop. So now you can probably see my desktop. Do you see what do you see right now?
It looks good. It's in like presenter mode.
Okay, great. And I'm going to have you over here on the right. Let me just see whether I can go back. There we go. Okay. So I'm going to stick the photo ribbon down there. Okay. So thank you very much. It's really nice to meet our 8P heroes and their parents and our fellow researchers that we're working with. So a few words about where my lab is. We're at UConn Health. We're about halfway between New York and Boston, which is where I trained. And it's a really great place to do stem cell research. We have a wonderful stem cell core. We have a great genome core for RNA sequencing, any type of genomic technology you can think of. We have a great single cell genomics core, but the thing I really want to highlight is that we have a really fantastic personnel for editing, for making very sophisticated iPSC, so induced pluripotent stem cell models for human genetic disorders. And we can make them transgenic and introduce tools like CRISPR and exist. So it's really wonderful. I'm very lucky to be in this place to do my research there. And today I'm really going to tell you about modeling chromosome dosage and correction of chromosome dosage in viable human aneuploidy. And then I'm going to switch towards CNV conditions towards the end. And if you're interested in our preprint, I put a QR code for our bioRxiv preprint for our Trisomy 21 work up there on the top right. So we're interested in viable human aneuploidy, which is really defined as the presence or the absence of a whole copy of a chromosome. But in addition to that, we're also interested in copy number variations, of which I've shown a few here. There's 8P, there's Prader-Willi and Angelman syndrome, various others. But by far the most frequent viable human aneuploidy is Down syndrome. And so it's a whole extra copy of chromosome 21. There's two other viable trisomies, Edwards and Patau syndrome. And we're also studying the absence of a whole chromosome. So that's when you don't have a second copy of the X chromosome or the Y chromosome. So you only have a single X. So that leads to Turner syndrome, as well as Klinefelter syndrome, when you have an extra copy of the X chromosome in the presence of an XY karyotype. So you're XXY. And so relative to some of the other disorders, to Edwards and Patau syndrome, and even Down syndrome, Klinefelter syndrome is relatively mild. And one of the reasons for that is that the X chromosome is rather special because it has a built-in system to count the number of chromosomes per cell and keep only one X active in the cell. So that means even if you're born with 3 X chromosomes or 4 or 5, the system will turn on all the additional copies of the X, will turn off all the additional copies of the X chromosome, so that in the end, you only are expressing a single X chromosome. And the reason for that process, which is called X chromosome inactivation, is that evolution came up with this process to essentially balance the 2 X chromosomes, the gene dosage from 2 X chromosomes in females and the single X chromosomes in males. And it does so by silencing that additional copy. But because it has that built-in counting mechanism, it will silence all additional X chromosomes beyond the first one, right? And so the way it does that is that it's a it makes a long non-coding RNA. So that just means it doesn't leave the nucleus. It doesn't code for protein. It spreads out over the second X chromosome where it's transcribed, where it's made. And then it basically kicks out the enzyme that transcribes all the other genes and recruits repressive chromatin marks that condense the chromosome and keep it locked up in the cell for the lifetime of the female, right? So all females have an inactive X chromosome, and this is the mechanism that ensures that that X chromosome remains inactive. And so our colleague, Jeannie Lawrence, had the great idea to try to translate this system to that extra copy of chromosome 21 and trisomy 21. And so what she did was she inserted a DOX-inducible, so an antibiotic inducible, so a small molecule inducible version of Exist, and put it on that third copy of chromosome 21. And so when you add that small molecule, it's called doxycycline, or DOX for short. What it does is it's transcribed on that chromosome, it spreads over that chromosome, and then locks it down, silences it. So the chromosome is still there, but the genes on that chromosome are no longer expressed, except for Exist, right? And the way that this works, you can appreciate here. So here we're looking at a cell nucleus. We're doing a technique called nascent RNA FISH, so we can see the 3 copies of a given gene. So in this case, the APP gene, which is thought to contribute to high rates of Alzheimer's disease in Down syndrome. And within 3 weeks of adding that small molecule doxycycline, you see a red cloud of Exist forming around that copy, but there is no longer a third copy of APP expressed. So you've gone from 3 active copies of APP to 2, right? So you've silenced 1 copy, and you've essentially returned to what we would call a euploid gene dosage. So even though there are 3 copies of the chromosome present, only 2 are active. The other one is inert and just sits in the cell. And so that was really great, a really great system. But the one thing that it didn't allow us to do was to take these, what's really great about human-induced pluripotent stem cells is that you can differentiate them to any cell type in the body, right? And you can study how that cell type might be affected by a mutation or by a copy number variant. But let's say you want to study neurons and you want to understand how trisomy 21 impacts neurons. Unfortunately, this system couldn't be turned on anymore once the cells were differentiated to neurons. So that's a technical limitation that allowed that system to not go as far as one would hope. And so we've tried to improve on that. So what I'm going to tell you today is that we really like to work with cells from mosaicism, so from mosaic patients, because it allows us to make iPSCs from the same donor that are otherwise genetically identical, but differ only in this case, the number of chromosome 21 copies. In trisomy 21, the way it works is that the embryo starts out in all likelihood trisomic, and then there's a second mitotic error that leads to loss of that third copy of chromosome 21. And that way we have fibroblasts that we can reprogram to euploid, so disomic and trisomic 21 iPSCs. So every time you see T21, that's the trisomic line. Every time you see D21, it's the euploid line. And we take our iPSCs through a battery of quality control checks after reprogramming. So we do a qPCR screen for other aneuploidies that are common in reprogramming, because reprogramming for the fibroblast to turn into a stem cell is a stressful process. And so sometimes they find a shortcut by duplicating another part of the chromosome or removing a part of a chromosome that allows them to grow better. And so one of the first things we do is we screen for the very common chromosomal changes that are sometimes enriched during reprogramming. Then we check that the cells are still pluripotent. We also do a genome-wide screen for any other segmental deletions or duplications. And then we go one step further and we sequence the whole genome. And the reason and the way we do that is it allows us to actually distinguish the maternal from the paternal copies in that individual. And then finally, we test their competence for neuronal differentiation. And so that gives us a really nicely controlled model where we can compare euploid and trisomic iPSCs, and we can differentiate them to neurons and compare those neurons as well. But I want to point out that that's really what I call a static comparison, because the trisomic iPSC went through neuronal differentiation as a trisomy, and the euploid one as a euploid cell. And so we don't really know that red question mark, we don't really know what happened in between, right? And so we also don't really know what a dynamic rescue would look like. So taking a neuron that is trisomic and then expressing exists to rescue its expression and correct it to a euploid expression profile. So that motivated our work. And so when we essentially we went through the same steps, we identified a variant that we could target with CRISPR, we integrated our copy of exist, and when we add doxycycline, these are male cells, so they don't have any endogenous exist, so they're not female, they don't have an inactive X chromosome. So the only exist clouds in red that you see here are coming from chromosome 21, where we put exist. And it recruits these repressive chromatin marks, H2A ubiquitin and H3K27 trimethylation. And it does so very reproducibly across 3 different lines and many rounds of experiments. And what we can also do is we can withdraw doxycycline, so we can express exist for a while and then take it off and see. And here I'm just showing you that when we take doxycycline off, so that's the WD, the withdrawal condition, exist is no longer expressed, right? So it's not expressed, it is expressed in plus dox, and then it goes away. And the reason I'm showing you this is because exist is actually initiating a cascade of changes on that chromosome that lock it down for good. So even once exist is lost, that chromosome is meant to stay transcriptionally inert. And so that's what I'm showing you here. We turn to RNA-seq to measure the gene expression changes across the chromosome. So in the top panel, I'm showing you all the chromosomes, and we have this on a log2 scale. So log2 just means we've transformed the data, but in essence, we're trying to go from 3 copies down to 2, right? So it's a reduction by 33%, or in log2 space, that would be minus 0.6. So that's where that dashed vertical line is. And we see here the distribution of our plus docs and our withdrawal samples. And even after we withdraw exist, those cells are expressing chromosome 21 as if there were only 2 copies in there. And on the bottom panel, I'm showing you that it doesn't matter where the genes are that we measure relative to where we integrated exist, which is this vertical line here. So we put it smack dab in the middle of the chromosome, and genes to the left and right are equally impacted. So they're all coming down to a minus 0.6 level, regardless whether they're close to the integration site or far away, right? So it's a very, very powerful and useful tool. And here's the third way in which I'm going to show you that, that the third copy is repressed. So I told you that we identified the variants that distinguish the maternal and paternal copies. And the 2 maternal copies are, or presumptive maternal copies, are nearly identical, and the paternal copy is a little bit different. So here we're just coloring them in 2 colors, in blue and orange. And in the euploid line, they're 50-50 because you get equal expression from both copies. But then in the trisomic line, as well as a trisomic line that's equipped with exist, but we haven't added dox yet, there is more of the blue allele. So the blue allele makes up about 2/3 of the expression. The orange allele makes up about 1/3 of the expression. Each one of these bars is a gene on the chromosome. And then when we add doxycycline, that orange bar gets really repressed, right? So now we really only have representation largely from the 2 maternal, from the blue copies. And that repression persists even after we withdraw exist or we withdraw doxycycline. There's some orange bars remaining, but those are probably technical artifacts because when we look at those corresponding genes, they're also reduced by that 1/3 level, that minus 0.6 log 2 fold change. So the exist-bearing third copy of chromosome 21 is silenced irreversibly. And now we're looking beyond the chromosome. There's lots of gene expression changes on other chromosomes because the genome responds to the overexpression from chromosome 21. And so here we're looking at the log 2 fold change or the change in these genes as it correlates with the difference between euploid and trisomic iPSCs. And so if they're correlated, they should fall along a diagonal, and they do. They fall along a descending diagonal, so they're anti-correlated. So another way of saying this is that the genes that were not on chromosome 21, but elsewhere in the genome, the genes that were overexpressed in trisomy 21 are repressed now, and the genes that were underexpressed in trisomy 21 are coming back up, right? So, and that's true regardless whether we look at the plus dox condition or the withdrawal condition. And then we can ask, okay, what biological processes are we impacting with this, what we call a rescue, right? And this is a very high level kind of plot that just gives you some themes. And in essence, we're seeing that in trisomy 21, we have an enrichment for chromosome 21 genes, of course, right? And so those are being repressed when we add dox, right? When we rescue the cells. And the same is true. So we have overabundant terms related to apoptosis and stress, and those are coming back up. And things that are repressed in trisomy 21 in Down syndrome, mitochondria, which generate energy, and the ribosomes that translate all proteins, right? Those are actually repressed in trisomy 21. They're blue, and they're coming back up when we rescue with exist dosage correction. This is another high-level plot just showing you, instead of the genes, correlation of the genes, I'm just showing you the correlation of gene term enrichments. And again, they fall on a descending diagonal when we compare trisomic to euploid expression on the y-axis to the change in expression or the change in enrichment on the x-axis when we add doxycycline. And many of those changes are actually preserved in neural stem cells or in neural lineages. So this is kind of a big summary because I wanted to save some time to actually talk about AP. But the reason I'm showing you this is because I mentioned the other trisomies, Edwards and Patau syndrome, and a lot of the pathways that I mentioned that generally fall in a category of proteostatic stress are also triggered by trisomies 13 and 18. And so that's led to the thought, well, the genes on chromosome 13 and 18 are not the same as the genes on chromosome 21. So if it's impacting similar pathways, maybe it's not because of the genes, but because of the mere presence of the chromosome, the extra chromosome in the cells. But if that was the case, we're not rescuing the extra copy is still there. So this distinguishing between gene-driven effects of the trisomy and aneuploidy-driven, so the mere presence of the chromosome, but not its expression, I think what our work shows is that because we can correct the overall gene dosage, not just on chromosome 21, but across the transcriptome, it really argues that this is not a real distinction, that essentially when you have a trisomy, if you can correct the gene dosage, you're also correcting these general stress pathways that can also be triggered by other trisomies like trisomy 13 and 18. And so in some ways that's good news because it means that, that there are cellular phenotypes that remain reversible because they depend on acute trisomy 21 overexpression, right? And it implies if we could rescue this overexpression, we could rescue these cellular phenotypes. So we then wanted to actually understand really how these cellular phenotypes, how stress might impact development. And one of the things we noted was that when we take our iPSCs and we differentiate them to neurons, we use an undirected differentiation protocol that also gives rise to the support cells that nurture the neurons that are called astrocytes. And we label them in their respective markers. We should see lots of red because neurons are born first. And then we should start seeing astrocytes in green. But instead, what we see and that's what we see in the isogenic euploid controls. But in our trisomy 21 iPSCs on the bottom panel, you see much more green early on at this stage of differentiation. And so what that tells us is that in both iPSC panels from two unrelated patients, the trisomy is essentially leading to early astrogenesis. And this is what this looks like in a close-up at 10.5 weeks. So these are long differentiation time courses, right? And so here you see some nicely red neurons and green astrocytes, and you can kind of, even from this low resolution view that only has a few dozen cells in it, you can see that there's more green on the right than on the left. Okay, so what can we do about that? Well, we can first ask whether if we rescue trisomy 21 with exist, whether the cells still make the same lineage choices. So are they still more likely to become an astrocyte than a neuron? And so here on the left, we have our euploid controls, lots of red. In the middle, we have our trisomic lines in green, right? They make lots of astrocytes. But if we add doxycycline, so if we start expressing exist either at the iPSC stage or on the first day of differentiation, we actually suppress the green and we get back red neurons. So these clones on the right are derived from this one here, from this panel down here in the middle, in the lower middle. And we're essentially changing a lineage bias from astrogenesis to neurons simply by either pre-silencing the chromosome or silencing it before the cells become neural progenitor cells. And so here I'm just showing you a close-up so you can appreciate the little dot in the cell that marks the H2A ubiquitin, that marks the silenced copy of chromosome 21. Okay, so then we counted this. We wanted to quantify this to see that this is reproducible. So this is the same data just in a bar format or in a box plot format. So you see more neurons in the euploid and fewer astrocytes, and you see a flip in the trisomic line. In the transgenic trisomic line, that carry exists, but we haven't added doxycycline yet. Just like their parental line, they make more astrocytes and fewer neurons. But when we take those exact same clones and we pre-treat them with doxycycline, or we add doxycycline on day 0, let them differentiate, now they make neurons, right? Of course, this doesn't work forever. First of all, it makes sense because neurons and astrocytes are actually born from the same neural stem cell. Those neural stem cells divide just to self-renew. And when they differentiate, they exit the cell cycle and they become neuron or astrocyte. And they typically become neurons earlier and then tend to bias towards astrogenesis later. And so what we see is a rescue of neurogenesis, which previously was suppressed at the expense, or we see a rescue of excess estrogenesis that was caused at the expense of neurogenesis. Now, of course, if we don't add doxycycline until the neural progenitor stage, then it's probably too late, right? Because the cells have already made that decision and we can't rescue them anymore. Okay, so the summary of this second part of the talk is something that I'm framing in the context of what was previously known, which is, sorry, I'm going to have to move the photo ribbon down here, the video ribbon. Okay, is that neural stem cells make neurons first and astrocytes later. And in Down syndrome, you have early and excess estrogenesis. And we're not the first group to show this. There's actually other groups that showed previously in either mouse models or even fetal samples that there's early and excess estrogenesis. What we're showing here is that it's very likely a primary choice rather than a selection, and that restoring the right gene dosage also restores the right differentiation potential of these cells. Now, we're also interested in long-term effects on the trisomy. Oh, I'm sorry. I see that I just want to make sure I don't miss any questions. Okay. If there's any questions, just feel free to interrupt me because I can't put the chat window up at the same time. So we're also interested in long-term effects on these cells. And so again, we have a strong correlation between what happens in iPSCs and what happens in neural lineages with trisomy 21. And one of the things we want to look at was apoptosis, which is cell death. And so we collaborated with Steve Finkbeiner's group at UCSF and sent his lab our lines because they had developed a really nice apoptosis reporter. The problem with tracking cell death is that once a cell dies, it's no longer there. It disintegrates very quickly. So you either need to track it very closely, or you need to get the cell to tell you that it's in the process of dying before it's gone. And that's exactly what Steve's group did. They developed a fluorescent reporter where the cells light up in red right before they die. And that's extremely useful because it allowed us to show that our trisomy 21 iPS-derived neurons were dying at a higher rate than the euploid and otherwise isogenic counterparts. So there's lots more other phenotypes that we can compare between euploid and trisomic iPS-derived neurons. But I want to get back to this experiment that I kind of set up at the beginning of the talk. What we really would like to understand in neurons is which cellular phenotypes, which neuronal phenotypes remain correctable once differentiation or development has finished. And so for that, we would need to be able to start with 3 active alleles, add doxycycline in neurons, and then turn one of them off. And then we could measure gene expression and neuronal and astroglial function and all these other cellular phenotypes that we are interested in that we think are therapeutically relevant. So another way of asking the same question is which phenotypes, cell types, and developmental windows remain amenable to therapy? And again, it requires exist to be induced in terminally differentiated neurons and astrocytes, which previously was not possible. So with our system, the way we wanted to approach this was we wanted to first show that we have terminally differentiated cells. And so for that, we took neural stem cells. So we differentiated our iPSCs for 3 weeks. So we had neural stem cells. Then we treated them with a compound called DAPT, which essentially forces the cells out of the cell cycle and into terminal differentiation. And we labeled the cells with a green analog, nucleotide analog that would turn the cells green. So we would know it would only turn the cells green if they were still dividing. So we could look at the number of green cells and know that we didn't have too many that escaped our DAPT treatment that were able to continue dividing. And then we did FISH or IF. So we measured either the abundance of the exist RNA or the abundance of the repressive chromatin marks at 6 and 10 weeks. And these conditions are referred to as day 0 plus dox if we added with doxycycline from the onset of differentiation, or PDAPT for post-DAPT when we would add doxycycline only after DAPT treatment. And so here I'm showing you that our DAPT treatment worked very well. We have very few green cells. This field of view was actually the most green cells we saw. So for the most part, the cells are really out of the cell cycle, and yet they're able to turn exist on when we add exist in the post-DAPT condition. And here I'm showing you the MAP2 and S100 beta staining to label the neurons and astrocytes, and the H2A ubiquitin is in white. And so you can see that we have some nice H2A ubiquitin foci, regardless whether we added doxycycline from day 0 or from only after DAPT treatment. So that tells us that we can induce exist in terminal neural lineages, but is it effective? So one thing that argues that is effective is, of course, the H2A ubiquitin staining, because it tells you that this chromosome acquired more of this repressive chromatin mark than the genome-wide background. That's why you can see a little dot in the cell. And we counted these. And so we were able to show that while it's not 100%, we have anywhere between 20 and 50% of the cells that turn on exist and that can recruit this repressive chromatin mark. Now, because it wasn't a pure population, because there were some cells that didn't express exist, we had to turn to single nuclear RNA sequencing to convince ourselves that we're actually rescuing trisomy 21 gene dose. But I want to get to 8p, so I'll just summarize real quickly. I showed you in iPSCs that exist silencing of trisomy 21 is near complete and irreversible in iPSCs. It reverses transcriptional hallmarks of cellular stress. And as long as we can silence the third copy of chromosome 21 before the neural progenitor stage, we can actually rescue this excess trisomy 21 estrogenesis that most likely is adaptive to protect neurons. But we can rescue it if we remove the need for it, for that adaptive change. And so we think that this astrogenic bias is due to primary skew, not selection. But we are aware that there are long-term consequences to those neurons that reduce their overall survival as well. And then lastly, I showed you that post-differentiation, we can also rescue trisomy 21 overexpression. So exist dosage corrects terminal post-mitotic neurons and astrocytes. And we can use that information to actually start to understand which trisomy 21 changes might be due to changes in neurons as opposed to astrocytes. And the reason that might be important is because it can tell us, for example, if we only rescue trisomy 21 in neurons, or we only rescue trisomy 21 in astrocytes in a given experiment, it might actually tell us what we can hope for if our therapies target astrocytes instead of neurons, because there might be some benefits to targeting astrocytes. And then the other benefit of this work could be that we could use exist as an in vitro benchmark for various drug candidates, because essentially it sets the gold standard for what is the best that you can hope for with a therapeutic approach, and compare that to a given drug candidate and see what aspects of the disorder or of the gene expression changes or of the cellular phenotypes it can rescue. The final way in which we are using exist, that we're exploiting it, is to validate cell stress reporters for screening. And I want to mention this because I think this could be relevant to 8p as well. So we have in these lines, we have exist integrate on that third copy of chromosome 21. And due to really great work from other labs that showed us, that informed us about the proteostatic stress and the exact mechanism of the oxidative stress in those cells, we're able to build a fluorescent reporter that would basically light up when the cells were stressed. And the way that it works is that here I'm showing you two iPS cell colonies that are not dox treated yet, so they're just trisomic, and they show you a certain level of stress, and we can fact sort these cells as well. And then when we add doxycycline and we silence that third copy of chromosome 21, we're actually losing expression of the oxidative stress reporter. So it actually drops by quite a bit. And I want to remind you that the reporter is not on chromosome 21, it's on 13, right? So we're not directly silencing the reporter. We're essentially reducing the amount of oxidative stress in these cells. And that's why the reporter is dropping off. So this could be really useful for screening, for example, to try to understand which genes on chromosome 21 are most contributing to oxidative stress in these cells. Now, with exist, unfortunately, we hit the whole chromosome, right? So we can use it to identify which cellular phenotypes might come from neurons or might come from astrocytes. But we can't really use it to distinguish whether it comes from gene A or gene B or gene C, right? So, but we could easily deploy this reporter in a CRISPR screening approach and delete different genes on chromosome 21 to see to which extent they rescue oxidative stress. And so that's how we hope we can move trisomy 21 research forward and link specific genes on that chromosome to oxidative stress, which I think is going to be therapeutically relevant. Now, chromosome 21 is a large chromosome with lots of genes. And so this is this is going to be a big undertaking. But chromosome 8p, especially the deletions, are much more, we're talking about a much smaller number of genes. And so I think what the other technology that I think helps us is that we have the variants identified across the chromosome that distinguish the paternal and maternal alleles. And so on chromosome 21, that meant that we could identify where they differed. We could measure gene expression in an allele-specific fashion, so we knew how much was this gene expressed from the two maternal copies versus the single paternal copy. We could also target exists to a specific copy that we picked beforehand. And we have all of these variants now that we can target to either increase or decrease gene expression. And we can target them at the chromatin level, or we can target them at the RNA level. And the only reason I mention that is because there's new RNA targeting CRISPRs in development that might be even therapeutically sensible, because at least they can be packaged into an AAV, which is not possible with currently with CRISPRa and CRISPRa. But at the moment, I mostly see the value of these tools as research tools. But that is not to say that the RNA CRISPRs are not already being used in vivo. So they're already being deployed in animal models of various disorders. And I'm just giving you some examples here. And there's still a lot of development and very fast development in this field. And I think our role is not so much to develop the tools, to be tool builders, but rather develop tools that we can deploy on a specific disorder so we can start understanding its biology. And so that brings me to chromosome 8p. I've replotted here some of the data from that very important paper from Okura et al. And so on the top, you see that short arm of chromosome 8. And just to remind you, most 8p cases are inf-dup-del cases that have an inversion of the central segment, a deletion of the distal segment, and a varying duplication along the rest of the short arm. And then there's also distal and proximal or long deletions that combine that two segments. And I just went through and I kind of wanted to tally up, try to get a better, like very low resolution view of the phenotypes, how they relate to these different copy number variants. And I categorized them broadly here according to table 1 in that paper. And I also put some median milestones up there to reiterate that the inf-dup-del cases are really the most affected, it appears. So I think the role that we can play in this very important problem is, I think we're getting to a point where Project 8p has done tremendous work funding the work that led to this really seminal paper. And so we're getting an idea of overall like low resolution genotype to phenotype correlations in patients. And I think what we can do now is we can build in vitro models, cell models, to identify cellular phenotypes. And we want to link those cellular phenotypes to developmental phenotypes, and then eventually link causative genes to these phenotypes. I think that's our best bet right now, because that will allow us to study the mechanisms, some pathway that is impaired, and then test interventions in vitro that might rescue that pathway. Once we have causative genes, we might actually be able to build an animal model, which we're currently lacking. So in orange is really where I see where our work falls. And the technical approach that we're taking, I kind of alluded to it already, is the CRISPR-I/CRISPR-A approach, because the benefit that it has is that instead of deleting genes, which can cause rearrangements, unexpected ones, we're just repressing or activating genes, right? And so we can combine this could be combined with a drug screening, or it could be combined with a fluorescent reporter screening. or it could be combined with single cell RNA sequencing. We're currently testing, so we've already built a euploid line that has all these components integrated into the genome. And we picked a system that actually allows us to target multiple genes at the same time. And we combined it with an inducible NGN2 system. So we could make neurons not the way we've been doing it over 10 and a half, 12 weeks, but in 10 and a half days or 8 days or 12 days, right? And so we think that's really going to speed up the discovery process. And it's very similar to the approach that Rarebase is taking, as you heard from Joe just the other month. So conceptually, I just want to make sure I don't have any questions, right? I'm almost done. Conceptually, what we could do is we could start from an INF-dup-del iPSC line and go top down. So in red, I mark the deletion. In blue, I mark the duplication, and the inverted segment stays gray. And if we targeted CRISPR-I and CRISPR-A, if we could do it in the same line, right, we could even try to, revert 8p back to a euploid state by repressing the duplicated genes and activating the copy of the genes that are deleted in 8p, right? Alternatively, we can start from a euploid line and target CRISPR-I and CRISPR-A to a euploid line and recapitulate 8p from the bottom up. So what is the minimal segment, like what's the minimal group of genes that we have to, for example, repress to get a similar cellular phenotype as what we're seeing in our INF-dup-del iPSCs? And so that way we could actually start dissecting within one genotype what would come out to be a distal deletion from a long deletion from a duplication only, right? And that way recapitulate bottom up what an INF-dup-del looks like. So in order to guide our work a little bit, I looked at the RNA-seq data that Joe Bellucci very kindly shared from Rarebase. And so just to remind you, what he presented was the NGN2 derived, the NGN2 driven neurons derived from iPSCs via NGN2 expression. And I didn't do much. I just took those gene expression results and looked for enrichment of specific gene terms. And so here I'm showing you something that came up in the discussion, which is that you see very nice enrichment of gene terms relating to chromosomal bands that are impacted by the 8p INF-dup-del case itself. So you see an upregulation of these duplicated segments. Those are the those are the blue arrows. And you see a downregulation of that distal segment that's deleted. So that makes perfect sense. So that's, so the data are really nice. We also saw that, and Joe showed this in his talk, that there's an upregulation of genes on chromosome 20. And so here I'm just showing you that this is also statistically significant, that there is an enrichment of genes on chromosome 20 that are upregulated. And of course, there's genes on other chromosomes that respond to chromosome 8p copy number, but this is probably something different, right? Because these genes are not distributed randomly, but they're concentrated on chromosome 20. And so one of the things that happens during iPSC reprogramming, and I alluded to this earlier, is that you're essentially challenging the cells to reprogram their genome to a pluripotent state. And some cells do it better than others, and you tend to select for rearrangements. And that's not a problem because you can screen them out. So it just means you have to screen more colonies when you reprogram iPSCs. And so we've done that. We've had in every single, when we reprogram from Turner syndrome and Down syndrome, even euploid cells, we've always had some clones we could not continue working with for that reason, because these are very common reprogramming associated artifacts. And we always had colonies that we could continue working with that did not have anything outside the chromosome that we were most interested in. So chromosome 20, unfortunately, is that duplication is one of the most frequent ones. But we have Project AP has generated many more iPS cell clones. And so we just have to screen them and pick the right ones to continue on this. Now, the other thing that I thought was encouraging was that I looked at the human phenotype ontology in their NGN2 data. And there's some things in there, I didn't select this, so there wasn't very much that was significant. So I'm showing you everything, and it's borderline significant. But there were gene terms that actually kind of made sense for 8p. And those included various seizure terms, as well as refractory problems and rare respiratory distress. And there are also some gene terms relating to serotonin and anxiety. So it doesn't mean that there isn't useful information in those experiments, in those data, right? It just means that we're probably going to get an even cleaner picture if we have iPS cell clones without that chromosome 20 duplication. Okay, so with that, I want to thank you for your attention. I want to thank the people that did the work, Aaron Banda, Heather Glad-Deely, and Prakar Bansal in my lab. I want to thank Lumine IDSC for the support of our Down syndrome work, and NIGMS for the support of our EXIST work, and of course, Project 8p for getting us started in 8p CNV disorder. And with that, I'd be happy to take your questions.
Thanks
Awesome.
very
Thank
much.
you.
Sure.
Anyone wanting to go first? Also, I know we had some questions in the chat, but want to open it up first for any live questions, and then Whitney, if we want to go back into that chat.
I see some great questions there. Shall I start?
Sure.
Okay. So Bina asked, on behalf of families, what is early astrogenesis? So like I mentioned, our neural stem cells, they make neurons first. So they self-renew, and then when they differentiate, they make neurons, right? But at some point, once they've made enough neurons, now they need to start making the cells that are going to support the neurons, that are going to mature the synapses, that are going to feed the neurons. And those are called astrocytes. And so those are made only after you've started making lots of neurons. Then you start making the support cells, the astrocytes. So when astrogenesis happens early, when the neural stem cell switches to making astrocytes early, it means that in essence, it'll make fewer neurons overall, and it'll make more astrocytes. In some ways, that can be really good because you're actually providing the support that those neurons need. On the other hand, you're not making enough neurons, right? Does that answer your question?
Yeah, thanks. I was just
Okay.
trying to think through as you were explaining it, if I didn't know what any of that meant, right? How is that relevant? And is that, I guess my follow-up question to that is, are there disorders that's clear and what that actually means in the human?
So I was actually already looking at Laura's comment, which is kind of related to that, right?
Right.
Because in essence, what I'm describing, this switch from neurogenesis to astrogenesis, it happens in utero, right? So it happens during development. It happens when mothers carrying an 8p pregnancy don't know that yet, right? So that even if we did develop something for in utero therapy one day, we would also have to develop mechanisms of testing for it early on, right? So I like to use that example as to highlight this is a developmental change that is already baked in. This is something we cannot revert after birth, right? But it's still important to understand the interplay between neurons and astrocytes because it can tell us about what cell type our therapies would have to target. And one of the interesting things about the interplay between neurons and astrocytes is that astrocytes are involved in clearing APP and clearing amyloid beta, which is a main driver of early Alzheimer's disease and Down syndrome. And it turns out that people with Down syndrome, even though they have really high rates of Alzheimer's disease, their rates are slightly lower and their onset slightly later than people that only have a duplication of the APP gene. And so I think that's really important because it might mean that their extra astrocytes are actually delaying their Alzheimer's syndromes, their Alzheimer's symptoms. So I don't know that this is going to be relevant to 8P, but it is very relevant to Down syndrome, right? And there's going to be cellular phenotypes that are going to be comparable across conditions, like, for example, proteostatic stress. And that's where some of the reporters that we're building might be very useful. And sorry, so just to bring that home, so the developmental phenotypes, Laura is absolutely correct. Like, we're not going back through development and reverting something, right? We can't do that. But what we can do is understand the dysfunction well enough that we figure out what is still. So the developmental changes we can't change, but the cellular phenotypes, those we can still target, right? So proteostatic or oxidative stress, that might be a cellular phenotype we can still target and get a therapeutic benefit. But first, we need to understand the mechanism behind it. Great questions. Thank you.
So for many of you that are waiting to see more 8P data from the Pinter lab, the iPSCs are ready to ship. And the karyotype results will come in. Apparently they're delayed, but hopefully they'll be fine and just another way to validate, but we'll get them in the queue. Hopefully I will drive them personally over to you, Dr. Pinter, if I have to, but they're ready to ship.
That'd be great. We have another question. Faye.
Hi. I really enjoyed that. Thank you very much. I have to say, though, maybe I just didn't quite follow at the beginning, and I'm not sure if you went through it or not. But and tell me where I'm wrong, or if, tell me how I go. So
If I know.
I'm sure you do. So with exist, right? This is something that typically inactivates the third or second X chromosome, right?
Mm-hmm.
Okay.
Yes.
So, and you're using it as a tool in the cells to silence the extra copy of 21, right?
Yes.
Okay. So how are you getting it to switch from chromosome X to 21?
Okay, you're right. I did not explain that well. You're 100% right.
And then my second question, which
Yeah.
is like a tag on, right?
Yeah.
Is and what's your proposal? Would you be able to silence just the duplicated region of 8P?
Yeah. So great question. So I apologize for not explaining that better. So you can take the exist gene, essentially whittle it down to its essential parts, stick it in add a promoter to it so you can drive it with doxycycline, and then take that entire construct and stick it onto chromosome 21. So now it wasn't on chromosome 21 before, we put it there.
Yeah.
And the trick with exist is it always silences genes in cis. So that just means it silences genes on the same chromosome that it's sitting on, that it's expressed on. And so that's great because you're getting a very precise gene dosage reduction. As I showed you, you get exactly 1/3, you get that copy silenced. But the one downside is it works great for this whole chromosomal aneuploidy because we hit every single gene on the chromosome. But on 8P, you don't want to do that because the genes on the long arm haven't been duplicated, right?
Exactly.
So that's actually why we didn't propose using exist on 8P. That's why we decided it makes more sense to target the genes one by one and figure out what are the candidate genes individually and what are they contributing individually, rather than trying to. I don't see exist really as a therapeutic tool right now.
Yeah.
I see it as a great research tool.
Okay, great. Perfect. Thank you.
Sure.
Well, I think we're up on time here. So if there's no other questions, then I think this was very informative. Obviously, we are all very much now excited for the 8P data to come, and we know that there were cell processing issues that were kind of creating some unexpected delays there. But but yeah, we are definitely very excited now to see the AP results.
Thank you, everyone.
All right,
Thank
well, with that, we'll wrap up. Mina, any last thoughts here?
No, I just wanted to say thank you so much, Stefan. Like this is, it's really helpful, and I think it's teaching us a lot about the method to the madness, right? Like it's very complicated. It's cutting edge advanced tools. And how do you apply that to our various disorders and what that really means, right? What's the conceptual framework for therapeutics? And it's really hard to make that connection, but we're starting to understand it better. So thank you so much for spending the time and presenting this all to us.
My pleasure. Thank you.
Thank you. Take
Thanks
care.
again. Have a good day, everybody.
Bye.
Thanks. Bye-bye.